Thanks for the introduction. So hello, everyone. Today I'll be introducing some of the concepts and methods that have been used for rare variant interpretation and also some of the applications in using rare variant interpretation in rare variant association studies. Okay, so in this presentation, there will be three main sections. Firstly, I will go through a brief introduction about most of the elements that are going to be covered here. And then there will be followed by a slightly heavier focus on rare variant annotation. And in the end, I will share some of our work that has applied these methods or tools in improving our understanding of rare variant and also population genetics through rare variant interpretation. First and foremost, rare variant plays a very important part in the genetic basis of human complex traits. In the recent years, there has been large-scale exome and genome sequencing discovering rare variants, which has provided this new opportunity for us to identify some causal mechanisms of diseases. So there has been decades of GWAS, which has successfully identified so many robust loci for common diseases. But this is still requiring us to do some additional work to get into the causal factors for the diseases. And at the same time, Mendelian disease study have also discovered high impact rare variants for rare diseases. But it is also curious to us, how would the rare variant contribute to those common diseases? We know that we can do something about this, but we still haven't fully explored this systematically yet. But for example, in some of the diseases such as schizophrenia, we have already established some early insights for rare variants that implicate dozens of loci, so that we have beginning to fill out this missing spectrum of rare or like intermediate frequency variants that contributes to other diseases of our interest. So just as a recap, when we're trying to understand how common variants impact the disease, we run a GWAS, which simply just pick a SNP, and then we run the statistical test to compare its frequency between the difference between cases and controls for a given disease. But then how it is different for rare variants, and what are the challenges here? So those might be some of the key underlying questions that we would like to know. And most intuitively, common variants are defined to be those variants with an allele frequency of over 1%, while rare variants' allele frequency are generally below 1% or even lower. So such a rarity of these variants often makes it very challenging to determine their significance because of the lack of statistical power. So since they individually have low frequency in the population, association studies for rare variant often uses an aggregated analysis to assess the combined effect of multiple rare variants within a certain genomic region. Most often, we just pick a gene and groups the variants within it, and then test it combinedly to improve power. So in this way, we can increase the power to detect association that might have been missed by just looking at a single rare variant in isolation. But this also poses questions on what strategy we should use for grouping rare variants. Like when we're grouping variant, do we want to grouping a specific variant function, variants with the specific variant function? Or do we want to filter by certain type, frequency, or whatever other criteria that make the variants more close to each other? So because we're interested in understanding more impactful variants, and those variants are often deleterious and pose significance in clinical setting. So, and therefore, such a deleteriousness will increase the risk of diseases, which also is likely to reduce the productive fitness, and this will result in a stronger natural selection on this variant, makes it rare, and return us less statistical power to truly understand the important impact on disease of our interest. So to address this issue, there are several approaches. First of all, we can, okay. So first of all, we can choose to design some new statistical methods to increase the power to detect rare variants, which is often very difficult because it requires many more assumptions and also other background information. And then secondly, we can also choose to optimize data scaling and management to get ready for a larger sample size of about millions of samples for like future whole exome or whole genome sequencing data. And then thirdly, we can just do a better variant annotation. This is where variant interpretation comes. So we need more accurate and efficient variant interpretation to better group the variant and then boost power in a proper way. As a brief overview here, variant interpretation evaluates whether these rare variants are benign or pathogenic. And also, we kind of want to assess their potential impact on protein functions or gene expressions. For example, you would often see analysis on rare loss of function variants, which interprets gene functions, and also missense variants that changes protein by altering an outcome amino acid. With a proper variant interpretation, we then can adequately group our variants and analyze the contribution of these rare or low frequency variants to diseases. So therefore, one of our most important motivations for variant interpretation is here, which is that with the correct annotation, we could be able to conduct better powered variant association study and identify high impact variations, because rare variants tend to be more directly impacting on certain phenotypes, and with such a high impactful rare coding variants aggregated within the gene, this would particularly resolve some of the limitations of GWAS, including to get a better understanding of the protein coding variants and the gene mechanisms underlies the associations with common non-coding variants. And secondly, with those rare variant association results, we can decipher the genomic basis of diseases, which will facilitate the development of disease risk prediction, drug target development, and then other like treatment developments. Last but not the least, analyzing rare variants can also enhance our understanding in human evolution, population migration pattern, and genetic diversity, which is also of great significance. So there are several approaches that could help enhance our understanding of rare variants. The first and most widely adopted one is this prediction tools used to assess variant impact based on their features, including conservation, structure, and non-functional adaptations, which is more often referred to as variant annotation tools. So there are many effective annotation tools developed in the past decades, including Polyphony tool, Revel, and most recently Alpha Missense using deep learning, and etc. So I will talk about some of these in detail later in this presentation. And then aside from functional annotations, large-scale sequencing data can also help us understand the patterns and the rates of variation within human population. For example, NOMAD, which is short for Genome Aggregation Database, provides allele frequencies from diverse populations, helping us determine the rarity of variants. And such information can also facilitate identification of regions with genes that are intolerant of deleterious variations, which are more likely to be associated to diseases and will offer more clinical interest. And we could also pursue functional studies such as experimental assays to test the impact of variants on gene functions or their protein activities. So as well, we can we can also check out some database specialized for particular information on pathogenicity of genetic variants. As an example here, ClinVar is like a crucial resource for this. We can use it to understand the clinical significance of specific mutations. It aggregates information about clinical significance of genetic variants from submissions across multiple sources. And this is especially useful for identifying rare pathogenic variants where such data may not often be available. And today I will be mostly focused on the first two categories, which are the variant annotation tools and also the reference population database. So I want to ask the question, what does variant annotation do? And basically, it attempts to answer two questions. The first one is, what does a variant do? Like, how does this variant impact on a gene of, or on a protein, on the transcript? And then also more specifically, what is this molecular consequence of this variant? How does it affect a gene or transcript? And is it loss of function, gain of function, or neutral? And then it is also very challenging to implement variant annotations for specific reasons. I'm listing two here. The first is that with the different levels of penetrance, there might be a different impact of the same variant. For example, when sometimes you can see that there is a disruptive variant in some apparently healthy individuals, who doesn't have the disease to be expected. And also, because the variant is rare, this is a very intrinsic property of the variations. We are often lacked of statistical power to learn the most robust information about such rare variant. So for example, we often assume that loss of function variant within the same gene is doing the same thing. And then it sometimes works because we're lucky, we're in a good situation. For example, we do this for BRCA1, and then those loss of function variants within this gene tends to act the same. But this is not often the case. They sometimes might have a very different impact on the same gene or on the same molecular process that we're assessing. And also as a note here, annotation is a slightly overloaded term. So here, when I'm talking about annotation, we mean the functional annotation of the variant. So to recall from the previous sections, one of the major challenges of variant association study is the lack of power. And in order to increase the statistical power, the key underlying logic of that is to group variant with the same function to boost the statistical power. However, it's not so straightforward to find out a well-defined group of variants with the same functional impact, as shown on this figure. So a same variant can feed into different categories. For example, a missense variants can also present as a gain of function or loss of function one, and most of them can be neutral or having just a very low impact. And then given that variants can have diverse functions, the most intuitive way to use to make this happen is to just use the most disruptive variants, because they tend to have a very strong impact, and then it's kind of safe to assume that they are acting towards the same direction, because they essentially just interrupt the gene function, which will end up in the most like harmful consequence to the downstream molecular pathway. So therefore, by aggregating them by aggregating those confident loss of function variant, predicted loss of function variant, and also those loss of function like missense variants, we can increase the statistical power and also have more association identified. But this can also cause a little bit of problem, because in order to include those high confidence impactful missense variants, we need to have a very delicate tool to identify the magnitude of impact of those missense variants, but this is not often the case. So by doing this, there is a chance that you will add some neutral variant into the group and introduce some noise or bias. So to summarize here, this is the one we're looking for, loss of function, which causes a very strong impact, premature termination of a protein. And then those variants are particularly interested because they provide a clear evidence about gene function by completely or partially knocking out the gene's activity. And then given their strong impact, they tend to have the same functional direction on diseases, making it perfect to group them and run the aggregation test. And then this variant can lead to specific phenotypation effects, making them highly relevant for understanding the genetic basis of diseases. And so how do we do that? How do we actually tag those variants with their functional annotation? So here, this is a snapshot of web variant effect predictor tool from Ensemble, which is one of the most popular tool people use to do variant annotation. And then it predicts the functional effects of genetic variants, such as SNPs, insertions, deletions, and structural variants on genes, transcripts, and protein sequences, as well as regulatory regions. So it also compares variants to database of non-genomic features, annotating them with information on consequence types. So with those annotations, we can also further obtain a primary annotation used in rare variant association, including protein truncating variants, also as similar to loss of function variant, missense variant, and a synonymous variant. And we can later use other specialized tools to further refine the categories and split missense variant into damaging and non-damaging categories so that we can include some into the loss of function category, where we can just test a group of highly impactful missense variant like as an individual group. But however, its variant annotation is even harder than it sounds. So here is an example of a stop loss variant where the A is replaced by a G, resulting in an interruption into the stop codon. And a sequence variant here, at least one base of the terminator codon is changed, resulting in a longer transcript. So such variants are often mostly classified as a loss of function variant. But the thing can be even more complicated in real life because there is an insertion here there is an insertion happening, which by definition could have been a frame shift variation. And this insertion is interrupting a stop codon, which indicates this can also be a stop lost variant. But in fact, this variant does not change the sequencing at all, meaning that this is indeed a synonymous variant in reality. So this just illustrates us how complicated a variant annotation can be. And then it's also even harder to correctly conduct annotation for non-coding variants, which accounts for a huge proportion of the entire genome. As said here, most of the genome is not in the exome. And also non-coding regions can have certain regulatory effect and generally located in a very diffuse way. So both of them will lead to complication in directly assessing any functional impact of those non-coding variations. Besides web that I have just mentioned, there are a lot more effective annotation tools developed in the recent years, which I have listed here. This is definitely not a comprehensive list, but are some of the interesting ones. I should also clarify that those tools does not necessarily fit into the very same categories. Some of them are algorithms, some of them are metrics, but they are here because they kind of share the spirit in assessing the functional impact of variants. And then they are doing this from a slightly different perspective through some sort of different ways. So for example, Revel and CAD aggregate scores from multiple tools, and MPC adopts regional constraint information, and then Alpha Missense obviously uses deep learning. So they're doing different things, but they are all aiming at predicting the variant impact from some sort of ways. And then still, I won't be talking through all of them in this presentation, but I will describe a little bit more into detail for the 4 tools highlighted here. So first, let's look at MPC, which is short for missense badness, polyphony 2, and constraint. It is a score predicts missense variant deleteriousness based on the evolutionary conservation. So before MPC, most of the tools for missense variant annotation were mostly not considering information on regional constraint. So to better address the challenges in interpreting missense variant, for example, they might often be neutral and present position-dependent effects. MPC incorporates natural selection information. So with regional constraint defined from likelihood ratio test plus some additional variant level metrics, therefore they form a metric called missense badness, which describes the deleteriousness of amino acid substitutions when they occur in a missense restrict region. And a missense depletion score was also computed from the original constraint model. And then the combination of missense badness, polyphen 2, and constraint outperformed other combinations of tools. So therefore forms the final score, which becomes the MPC score. And in practical use, a variant with an MPC score over 2 indicates a high likelihood of being deleteriousness. And another way of looking at this is that we can also utilize transcript level information to infer functional impact of variants, because a single gene can produce multiple transcripts through alternative splicing, meaning that pre-mRNA produced from a gene can be spliced in different ways to include or exclude certain exons. So therefore, variants can have effect on one or more transcripts. The two ways we often use to interpret this is first one, the worst consequence, referring to the most severe predicted impact on a variant that has across all the possible transcripts of a gene. And the second one is the canonical transcript, which is often the longest and is used for consistency in analysis, serving as a standard reference. So this approach ensures that variant interpretation considers the most significant potential effect on gene function. It's also quite crucial for understanding genetic diseases and informing some clinical insights. So inspired by this phenomenon that an exon of a gene to be expressed at varying level across different cell types, another approach, PAXT, was developed to incorporating exon expression into variant interpretation. It gives each variant a score based on how much the variant affects the gene transcripts. So this helps determine the potential impact of a variant located within specific exon. And if an exon has a low expression level and harbors a variant, the variant might not have a very strong impact on that gene's overall function compared to the variant that are identified in a high expression region. So it is also being used to select predicate loss of function variants with extremely high impact from the highly expressed exons. And then this metric can also be applied into rare variant association test. So in this case, we not only just group variant into the primary functional groups, including loss of function, missense, and synonymous, but we also group them by their PAX expression level. So this figure here on the bottom presents the transcript expression-based analysis in patients with developmental disorder and autism. We can see that the loss of function variant found from the highly expressed regions have a larger effect than those in the weakly expressed regions. And also, notably, those loss of function variant found on the regions with little evidence for expression basically performs similar to those synonymous variants, which suggests that such a variant can be removed from analysis of gene burden testing. So it's serves as a complementary way of selecting our variants to be grouped into our variant association test. And here I'm introducing another metric called MAPS, which is inspired by another challenge for assessing deleteriousness. So as we know, the mutation rates differ across different types of variants like transitions, transversions, and it will affect the distribution in functional classes like synonymous and missense mutations. And this will also impact the proportion of variants that are singletons. So MAPS here was created to address this bias, which essentially to correct the metric, which is previously called a proportion singleton metric. So this is meant for a correction considering the expected singleton proportion for each classes. Essentially, this is a metric for rarity based on the fact that more constraint categories harbor a higher proportion of singleton variants. And then also as shown on the figure to the right, MAPS effectively distinguished between different functional groups and also corrected the misclassified low confidence loss of function variant to more of like a missense missense-like category. And then another, so the tools, yeah, so this is another tool which is called Lofty that basically focuses on loss of function variance. And it is a web plugin to identify loss of function variation. And then with certain filters and selections, it creates a high confidence set of loss of function, and then therefore removes loss of function annotation artifacts. So for example, it removes terminal truncation variants and rescues slice variants, and that are predicted to escape nonsense mediated decay. And currently, this tool focuses on variants from stop gain, the splice site disruption, and frame shifts variants. And it helps identify such variant and notation error by providing confidence level for predicted loss of function variants and further refine our selection on such high impact of ones. And here comes our new star, which has been under hot discussion for a while since the end of last year. So missense, alpha missense was developed by Google. It's one of the most recent and integrated deep learning model to understand the functional impact of missense variants. So basically, it uses population frequency data sequence and predicted structural context and then train those data through unsupervised protein language. The population data used here is from Nomad V2 and also the primate data. So by comparing the occurrence of variants in human and the primate data, one can effectively identify the deleteriousness of a variant. And then another key feature of alpha missense is that it is not only trained directly from sequences, but trained with AlphaFold, which has been pretty well established and trained from protein structure. So it has already had some decent background knowledge of the protein sequences, giving it a better chance to predict the consequences of missense variants and categorize their pathogenicity. So this is a short like a screenshot of how this algorithm works in a very simplified way. For anyone who's more interested in the deep learning algorithm, you can take a look at the paper. Yeah. And then after introducing all the variant annotation tools, here we're jumping to our second approach, which is using reference population database to interpret variants. We're now in the Nomad V4 era, which has a collection of about 800,000 samples from 8 major ancestry groups, which enable users to obtain a more reliable allele frequencies information and identify rare variants within diverse populations. It can also help us improve rare variant classification accuracy, enable rare disease genetic research, supports population-specific variant interpretation, and also serves as an essential tool, essential resource for rare variant functional impact studies. So analysis with Nomad have also pinpointed a number of loss of function variants that can alter protein function significantly, offering insights into genes that might tolerate loss of function in at least one individual. So such data is really crucial for identifying disease-causing mutations and understanding the human capacity for gene function loss. And also another thing to be noticed is that the resource has also been made into this great public browser that is very easy to use. You can interactively search for information of a gene, transcript, or variant of your interest, and also download the data for your research purpose. And many of the annotations I just mentioned today were also included in the Nomad release data. For example, we have Lofty, PAXT, VAP, and also CAD. They're all available in the public data, making our evaluation on variant annotations much easier. And for some applications of rare variant interpretation, here is one of the examples. So in 2022, we have published the result for GeneBAS, which is a systematic association analysis of the UK Biobank whole exome sequencing data, which include association test results for about 400,000 whole exome sequencing samples, and then across over 4,000 phenotypes. So as shown in the figure to the top right, grouping rare variants with allele frequency below 1%, we have conducted the rare variant association test for 3 different functional annotations and across all the genes. On the y-axis of that that figure on the top right, we are showing the proportion of genes with at least 1 significant associations across the 4,000 phenotypes. So within each allele frequency bin, the magnitude of this proportion aligns with the functional impact of annotation groups. And as we can see in each of the category, loss of function is more likely to have an impact on human traits, followed by missense, and then synonymous groups had barely any associations, with just a few on the more common end, which is more likely to be driven by correlation with nearby high impactful variants. And also biobanks like the UK Biobank provide information not only just on genotypes from a large scale cohort, but also thousands of phenotypes matched to those genotypes available. So here we are also categorizing the disease phenotypes based on their ICD chapter codes and was able to examine the rare variant associations within each disease domains. With such information, we can explore rare variant function from a broader perspective. By jointly evaluating their effect across diverse types of phenotypes, we can have a more comprehensive view of the potential underlying genetic basis and how they are shared or different across those phenotypes. And this result is also made into a public browser here, which you can take a look and pick your favorite gene or phenotype and explore the association results and the effect distributions yourself. And then another application is just like a broader version for rare variant association analysis. So the Biobank Rare Variant Analysis Consortium, formed in February 2022, is a collaboration now including 16 global biobanks and cohorts aimed at analyzing rare genetic variants. It is significant in leveraging the collective power of diverse biobanks to improve the understanding of rare genetic variants' impact on health and diseases. With such effort, we can not only increase statistical power of rare variant association, we also enable the cross-validation of results across biobanks and also analyze the understudied traits or diseases, and also enhance genetic diversity of rare variant association study, advance methodology, promote collaboration, and also in this case, aid in the identification of those significant genetic factors contribute to rare diseases and potentially discover some more therapeutic approaches with this global effort. Yeah, this is pretty much all for my today's presentation. For some future directions, we would hope to develop some better and more accurate interpretation tools for both coding and non-coding variants, obviously. And so that we can probably incorporate more relevant information in a more dynamic way. And then we also wanted to use more rare variant association study in more diverse population across diverse phenotypes. And then we are also started to thinking about effectively running RVAS on the non-coding variants, but we're still working on finding the good annotation approaches to make it powered enough. And here is an ongoing work of the All of Us data, where we run association testing for both common rare variants across thousands of phenotypes and across diverse ancestry groups, which I hope could help refine annotation metrics as well as promote our understanding of rare variants and their contributions to many more traits of our interest, and then can improve human health as well as facilitate treatment development. And yeah, above all, I would like to thank everyone here and also everyone who has contributed to all the projects that I've been working on, including the analysts, the data project management team, and also the participants to the studies. And specifically, I would like to thank my PIs, Ben and Conrad, for their contribution in the work, their continuous guidance and support, and also everyone from our labs and the NTGU community, whom I have the great honor to work with for the past 4 delightful years. And thanks for listening.